Welcome. This is a pre-class video for our flipped session on protein folding, unfolding, and misfolding with a focus on prion and prion-like diseases. Please watch it before we meet because in class you'll answer 8 USMLE Step 1 style questions built directly on what we cover here. The whole video runs about 25 minutes. Our goal is simple: to see how a single idea, a protein taking on the wrong shape, explains a surprising range of human disease, from Alzheimer's and Creutzfeldt-Jakob disease to type 2 diabetes and emphysema. Here are the 6 objectives for the session. First, you should be able to explain how a protein reaches its correct shape and how the cell disposes of proteins that don't. Second, describe what amyloid is and how it forms. Third, connect the major neurodegenerative diseases to protein aggregation and understand how those aggregates spread in a prion-like way. Fourth, explain the prion mechanism itself. Fifth, distinguish the different amyloidoses by the protein that forms them. And sixth, use alpha-1 antitrypsin deficiency as a model of ER misfolding disease, and understand the drugs that target proteostasis. Keep these 6 in mind. Every in-class question maps to one of them. Let's start with normal folding. The central principle from Anderson's classic experiment is that a protein's amino acid sequence contains all the information needed to specify its three-dimensional structure. The native functional fold is the conformation with the lowest free energy, and the chain funnels down towards it rather than searching randomly. That's the resolution of what is called Leventhal's paradox. So remember that, Leventhal's paradox. Local structure forms first and constrains everything that follows. On the slide, you see one clinically important nuance. The native state is only marginally more stable than the unfolded state. That small energy margin is a feature. It lets proteins flex and function, but it also means a mutation, chemical damage, or cellular stress can tip a protein towards misfolding. Think about that and hold on to that idea, because misfolding is the theme of everything that follows. Okay, so we're dealing now with chaperones. Cells don't leave folding to chance. They actually invest heavily in quality control. And there are 2 arms of this control. One on the left, the molecular chaperones, like HSP70, which bind the exposed hydrophobic stretches of an unfolded chain and prevent it from clumping with its neighbors. So you're preventing aggregation. Notice what they do and don't do. They buy the protein time and space to fold, but they do not supply the folding information. Remember that. Chaperones do not supply the folding information. That is in the sequence. Chaperonins, such as GROW, take this further, enclosing a single protein in a chamber to fold in isolation. On the right is the endoplasmic reticulum quality control. And this is response to failure. When misfolded proteins pile up, the unfolded protein response through IER1, PERK, and ATF6 raises folding capacity and slows new synthesis, while ERAD ships misfolded proteins out to the proteasome. Remember, the proteasome is for degradation. If the stress can't be resolved, the cell triggers apoptosis or cell death. And that's exactly what kills beta cells in type 2 diabetes and hepatocytes in alpha-1 antitrypsin deficiency. So what happens when folding fails and quality control is overwhelmed? Follow the arrow on the slide. A native, largely alpha helical protein misfolds into a beta sheet-rich form. Those misfolded units first assemble into small oligomers, which are actually the most toxic species, and then into the long insoluble amyloid fibril. The key structural feature is at the end, across beta core, where beta strands stack perpendicular to the fibril axis. Three things to remember. First, this is nucleation dependent. A slow seed forms and then growth is rapid, and those seeds can propagate. Second, every amyloid, no matter which protein it came from, shares that cross beta core, which is why they all bind Congo red and show apple green birefringence under polarized light. That's your universal histologic signature. And third, these fibrils are extraordinarily stable. They resist both chaperones and the proteasome, so deposition is slow, relentless, and largely irreversible. Amyloid aggregation is a shared thread through the big three neurodegenerative diseases. In Alzheimer's, two proteins misbehave. Amyloid beta, cleaved from the amyloid precursor protein, builds up as extracellular plaques, and hyperphosphorylation tau forms intracellular neurofibrillar tangles. In Parkinson's, the culprit is alpha-synuclein, which aggregates into intracellular Lewy bodies, producing the classic resting tremor, rigidity, and bradykinesia. In Huntington's, an expanded polyglutamine tract in the Huntington protein, the result of a CAG trinucleotide repeat, drives aggregation, giving chorea and dementia. And because the repeat can lengthen between generations, the phenomenon known as anticipation. Different proteins, different clinical pictures, but the same underlying event. A normal protein aggregating where it shouldn't. Now, here's the concept that ties these diseases to prions. These aggregates don't just sit there. They can template their own spread. A misfolded aggregate acts as a seed that converts nearby normal protein into the same misfolded shape, and the pathology then advances along connected neural pathways. This has been shown experimentally for amyloid beta, tau, and alpha-synuclein. Because this looks so much like what prions do, we call it prion-like. But be precise about the language. Prion-like means it spreads within a single brain by templating. It does not mean these diseases are contagious between people. Alzheimer's and Parkinson's are not transmittable the way a true prion disease can be. That distinction is a favorite exam point, so keep it sharp. That brings us to true prions, genuinely one of the strangest ideas in biology. A protein that changes another protein with no gene and no nucleic acid involvement. On the left is the normal cellular prion protein, PRPC, a normal, mostly alpha helical membrane anchored protein. On the right is the Scrapey's form, PRPSC, exactly the same amino acid sequence, but folded into a beta sheet-rich protease-resistant shape. The disease is nothing more than that shape change, and it's self-propagating. PRPSC acts as a template that forces normal PRPC to adopt the misfolded form. This is what is called the protein-only hypothesis. In the brain, it produces spongiform change, a vacuolated sponge-like appearance. And because PRP is a normal self-protein, there is no inflammatory response. Human prion disease comes in 3 flavors, and the exam wants you to recognize all 3. Sporadic Creutzfeldt-Jakob disease is by far the most common. PRPSC arises spontaneously, and it presents as a rapidly progressive dementia with myoclonus. Familial forms, familial CJD, Gershman, Straussler, Schrinker, and fatal familial insomnia come from inherited mutations in the PRNP gene. And acquired forms come from outside the body. Variant CJD from BSE infected beef, Kuru from ritual cannibalism, and iatrogenic cases from contaminated tissue. The diagnostic clues are in the box. RT-QULC and the 14-3-3 protein in the spinal fluid, cortical ribboning on diffusion MRI, and periodic sharp waves on EEG. Whatever the cause, prion disease is relentlessly progressive and invariably fatal. When amyloid deposits outside the nervous system, we group the diseases by the protein that forms them. And this table is high yield, so learn it by precursor. AL, amyloid, is made of immunoglobulin light chains, and it points you to a plasma cell problem like multiple myeloma. Classically, it hits the heart and kidneys and can cause macroglossia and periorbital purpura. AA amyloid is made of serum amyloid A, an acute phase reactant, so it follows chronic inflammation, rheumatoid arthritis, inflammatory bowel disease, chronic infection, and tends to affect the kidneys. ATTR is transthyretin, either a hereditary mutant form or the wild-type senile form, and it typically causes cardiomyopathy and polyneuropathy. The drug tafamidis is aimed right at it, and IAPP, or amylin, is a localized amyloid that deposits in the pancreatic islets in type 2 diabetes. One question on the memory hook. The precursor tells you the disease. Remember, the precursor tells you the disease. Alpha-1 antitrypsin deficiency is my favorite illustration of ER misfolding, because one molecular defect damages 2 organs in 2 completely different ways. On the left, the liver. The Z variant of the protein misfolds and polymerizes and gets stuck in the hepatocyte's endoplasmic reticulum. It can't be secreted. Okay, it's stuck in the ER. Those retained polymers show up as PAS positive, diastase-resistant globules on biopsy, and the ER stress they cause leads to cirrhosis. This is a gain of misfolding. The damage comes from protein that's trapped inside the cell. On the right, the lung, where the problem is the opposite, because the protein never gets secreted. There's almost none in the blood. So neutrophil elastase runs unopposed and destroys the alveolar walls, causing panacinar emphysema that's worst at the lung bases. The reverse of the upper lobe pattern you see with smoking. The vignette to lock in is at the bottom. A young non-smoker with basilar emphysema and unexplained liver disease. Check the serum level and the pi genotype. The ZZ genotype is the most severe. Understanding misfolding also explains a set of modern drugs grouped here by strategy. The first strategy is to block the proteasome. Bortezomib does this, and it works beautifully in multiple myeloma because myeloma plasma cells churn out enormous amounts of immunoglobulin and are utterly dependent on the proteasome to clear the misfolded excess. Take that away, and the toxic protein accumulates until the cell dies. The second strategy is to stabilize the protein so it can't misfold in the first place. Tafamidis binds and stabilizes the trans-therapin tetramer so it doesn't fall apart and aggregate, treating ATTR. And the third strategy is to rescue a misfolded protein trafficking. The CFTR modulators, correctors like lumacaftor and tezacaftor, plus the potentiator ivacaftor, recover the delta F508 CFTR protein that IRAD would otherwise destroy. Notice that this is the same ER quality control logic from earlier, now applied to a channel. Before we finish, here are 6 associations that map straight onto Step 1 questions. It's worth committing to memory. Apple green birefringence means amyloid of any precursor. A rapidly progressive dementia with myoclonus plus 1433 or RT-QULC means CJD. A young non-smoker with both emphysema and liver disease means alpha-1 antitrypsin deficiency. Macroglossia and periorbital purpura with a monoclonal protein means AL amyloidosis. Islet amyloid in the pancreas means type 2 diabetes from amylin, and bortezomib is a proteasome inhibitor used in multiple myeloma. If you know these 6 codes, you'll recognize most of what we do in class. That's the material. When we meet, you work through 8 one best answer questions built directly on this video. You'll be sorting the lookalikes the way the exam does. AL versus AA versus ATTR, amyloid. Sporadic versus variant CJD. And the liver versus lung effects of alpha-1 antitrypsin deficiency. Come ready to explain folding and quality control, amyloid and prion mechanisms, the neurodegenerative and prion-like diseases, the amyloidosis, and the therapeutics. If you'd like to read further, the Folding folding and misfolding chapters in Leninger. And Harper's cover all of this. See you in class.